in the name of throwing out kind of new unproven ideas, right? So this is this is just my conjecture. We've done some computational modeling of it, which I initially, this was a very clever student that I work with named Lakshwan, who did some models with me, and I initially thought it was a bug, and then I realized that no, actually, this is the feature. The idea is this: imagine. So we've been working for a long time on a concept of competency among embryonic parts, and what this means is basically the idea that there are homeostatic feedback loops among various cells and tissues and organs that attempt to reach specific outcomes in anatomical morphospace despite various perturbations. So the idea is that if you have a tadpole and you do something to it, whether by a mutation or by a drug or something, you do something to it where the eye is a little off kilter or the mouth is a little off, all of these organs pretty much know where they're supposed to be. They will try to minimize distance from other landmarks, and they will remodel, and eventually you get a normal frog, so that they will sort of recover the correct anatomy despite starting off in the wrong position, or even things like changes in the number of cells or the size of cells. They're really good at getting their job done despite various changes, right? So okay, so they have these competencies to optimize specific things like their position and structure and things like that. So that's competency. Now here's the interesting thing. Imagine that you have a species that has some degree of that competency. And so you've got an individual of that species, comes up for selection, fitness is high, looks pretty good, but here's the problem. Selection doesn't know whether the fitness is high because his genome was amazing, or the fitness is high because the genome was actually so-so, but the competency sort of made up for it, and now everything kind of got back to where it needs to go. So what the competency apparently does is shield information from evolution about the actual genome. It makes it harder to pick the best genomes because your individuals that perform well don't necessarily have the best genomes. What they do have is competency. So what happens in our simulations is that if you start off with even a little bit of that competency, evolution loses some power in selecting the best genomes, but where all the work tends to happen is increasing the competency. So then the competency goes up, so the cells are even better at, and the tissues are even better at getting the job done despite the bad genome. That makes it even worse. That makes it even harder for evolution to see the best genomes, which relieves some of the pressure on having a good genome, but it basically puts all the pressure on being really competent. So you've got this. So basically, what happens is that the genetic fitness basically levels out at a really suboptimal level, and in fact, the pressure is off of it, so it's tolerant to all kinds of craziness. But the competency and the mechanisms of competency get pushed up really high. So in many animals, but there are other factors that sort of push against this ratchet, but it becomes a positive feedback loop. It becomes a ratchet for optimal performance despite a suboptimal genome. And so in some animals, this sort of evens out at a particular point. But I think what happened in planaria is that this whole process ran away to its ultimate conclusion. The ultimate conclusion is the competency algorithm became so good that basically whatever the genome is, it's really good at creating and maintaining a proper worm because it is already being evolved in the presence of a genome whose quality we cannot control. So in computer science speak, it's kind of like, and Steve Frank put me onto this analogy, it's kind of like what happens in RAID arrays. When you have a nice RAID array where the software makes sure that you don't lose any data, the pressure is off to have really high quality media. And so now you can tolerate media with lots of mistakes because the software takes care of it in the RAID, and the architecture takes care of it. So basically, what happens is you've got this animal where that runaway feedback loop went so far that the algorithm is amazing, and it's been evolved specifically for the ability to do what it needs to do, even though the hardware is kind of crap. And it's incredibly tolerant. So this has a number of implications that to my knowledge have never been explained before. For example, in every other kind of animal, you can call a stock center and you can get mutants. So you can get mice with kinky kind of kinked tails, you can get flies with red eyes, and you can get chickens without toes, and you can, humans come with as various, albinos and things. There's always mutants that you can get. Planaria, there are no abnormal lines of planaria anywhere, except for the only exception is our two-headed line, and that one's not genetic. That one's bioelectric. So isn't it amazing that nobody has been able, despite 120 years of experiments with planaria, nobody has isolated a line of planaria that is anything other than a perfect planarian. And I think this is why. I think it's because they have been actually selected for being able to do what they need to do, despite the fact that the hardware is just very junky. And so that's my current take on it. And really, it puts more emphasis on the algorithm and the decision making among that cellular collective of what are we going to build, and what's the algorithm for making sure that we're all working to build the correct thing.
So if you translate this idea into computer science, a way to look at it is, imagine that you find some computers that have hard disks that are very, very noisy. And where the hard disk basically makes lots and lots of mistakes in encoding things, and bits often flip and so on. And you will find that these computers still work, and they work in pretty much the same way as the other computers that you have. And there is an orthodox sect of computer scientists that thinks it is necessary that every bit on the hard disk is completely reliable, or reliable to such a degree that you only have a mistake once every 100 trillion copies. And you can have an error correction code running on the hard disk at a low level that corrects this. And after some point, it doesn't become efficient anymore. So you need to have reliable hard disks to be able to have computers that work like this. But how would these other computers work? And it basically means that you create a virtual structure on top of the noisy structure that is correcting for whatever degree of uncertainty you have, or the degree of randomness that gets injected into your substrate. Dave Eckley has a very nice metaphor for this. You know him, maybe?
Yeah, I know Dave.
Yeah, he's I think, a beautiful artist who explores complexity by tinkering with computational models. I really find his work very inspiring. And he has this idea of best effort computing. So in his view, our own nervous system is a best effort computer. It's one that does not rely on the other neurons around you working perfectly. But make an effort to be better than random. And then you stack the improbabilities empirically by having a system that evolves to measure, in effect, the unreliability of its components. And then stack the probabilities until you get the system to be deterministic enough to do what you're doing with it, to do with it. Right? So you if you have a system that is, as in the planaria, inherently very noisy, where the genome is an unreliable witness of what should be done in the body, you just need to interpret it in a way that stacks the probabilities, that is evaluating things with much more error tolerance. And maybe this is always the case. Maybe there is a continuum, maybe not. It's also possible that there is some kind of phase shift where you switch from organisms with reliable genomes to organisms with noisy genomes, and you basically use a completely different way to construct the organism as a result. But it's a very interesting hypothesis then to see if this is a radical thing or a gradual thing that happens in all organisms to some degree.
Yeah.
What I also like about this description that you give about how the organism emerges, it maps on, in some sense, also in how perception works in our own mind. At the moment, machine learning is mostly focused on recognizing images, so or individual frames, and you feed in information frame by frame, and the information is relatively disconnected. A system like DALL-E 2 is trained by giving it several hundreds of millions of images. And they're disconnected. They're not adjacent images in the space of images. And a baby could not probably learn from giving 600 million images in a dark room and only looking at this and deduce the structure of the world from this, whereas DALL-E can, which gives testament to the power of our statistical methods and hardware that we have, that far surpasses, I think, the combined power and reliability of brains, which probably would not be able to integrate so much information over such a big distance. For us, the world is learnable because its adjacent frames are correlated. Basically, information gets preserved in the world through time. and we only need to learn the way in which the information gets transmogrified. And these transmogrifications of information means that we have a dynamic world in which the static image is an exception. The identity function is a special case of how the universe changes. And we mostly learn change. I just got visited by my cat, and my cat has difficulty to recognize static objects compared to moving objects, where it's much easier to see a moving ball than a ball that is lying still.
Yeah.
And it's because it's much easier to segment it out the environment when it moves, right? So the task of learning on a moving environment, a dynamic environment, is much easier because it imposes constraints on the world. And so how do we represent a moving world compared to a static world? The semantics of features changes. And an object is basically composed of features that can be objects themselves. And the scene is a decomposition of all the features that we see into a complete set of objects that explain the entirety of the scene and the interaction between them. And causality is the interaction between objects, right? And in a static image, these objects don't do anything. They don't interact with each other. They just stand in some kind of relationship that you need to infer, which is super difficult because you only have this static snapshot. And so the features are classifiers that tell you how to whether a feature is a hand or a foot or a pen or a sun or a flashlight or whatever, and how they relate to the larger scene, in which, again, you have a static relationship in which you need to classify the objects based on the features that contribute to them. And you need to find some kind of description where you interpret features, which are usually ambiguous and could be many different things, depending on the context in which you interpret them, into one optimal global configuration, right? But if the scene is moving, this changes a little bit. What happens now is that the features become operators. They're no longer classifiers that tell you how your internal state needs to change, how your world needs to change, how your simulation of the universe in your mind needs to change to track the sensory patterns, right? So a feature now is a change operator, a transformation. And the feature is, in some sense, a controller that tells you how the bits are moving in your local model of the universe. And they're organized in a hierarchy of controllers. And these controllers need to be turned on and off at the level of the scene. And they have a lot of flexibility once you have them. They can move around in the scene. They're basically now self-organizing, self-stabilizing entities. In the same way as the mouse is moving around in your organism, a feature can move around in the organism and shift itself around to communicate with other features until they negotiate a valid interpretation of reality.
That's incredibly interesting because, as soon as you started saying that, I was starting to think that the virtualization that enables, right? So the earlier part of what you were saying, the virtualization of the information that allows you to deal with unreliable hardware and everything. The bioelectric circuits that we deal with are a great candidate for that, because actually we see exactly that. We see a bioelectric pattern that is very resistant to changes in the details and make sure that everybody does the right thing under a wide range of different defects and so on. But even more than that, the other thing that what you were just emphasizing, the fact that we learn the delta, right, and that we're looking for change. Very interesting. If you pivot the whole thing from the temporal domain to the spatial domain, so in development, what we when we look at these bioelectric patterns, now these patterns are across space, not across time. So unlike in neuroscience, where everything is kind of in the temporal domain for neurons, these are static voltage patterns across tissue, right, across the whole thing. So for the longest time, we asked this question: how are these read out? How do cells actually read these? Because one possibility this was a very early hypothesis, 20 years ago, was that maybe the local voltage tells every cell what to be. So it's like a paint by numbers kind of thing, and each voltage, each voltage value corresponds to some kind of outcome. That turned out to be false. What we did find is that and we have computational models of how this works now. What is read out is the delta, the difference between regions. It doesn't care. Nobody cares about what the absolute voltage is. What is read out in terms of outcomes for downstream cell behavior, gene expression, all that. What is actually read out is the voltage difference between two adjacent domains. So that is exactly actually what it's doing, just in the spatial domain. It only keys off of the delta. And what is learned from that is exactly just as you were saying, it modifies the controller. for what's downstream of that. And there may be multiple ones that are sort of moving around and co-inhabiting. I mean, it's a very compelling picture, actually, and way to look at some of the simulations that we've been doing about how the bioelectric data are interpreted by the rest of the cells.
If you enjoyed this toe clipping, then the full video is linked in the description. You should also sign up for Toe Mail, which is again in the description and the pinned comment. You'll receive immediate access to all exclusive updates from the Theories of Everything podcast. It also helps me communicate directly with the core toe community. You'll also receive my top 10 toes. Think of it as the intellectual version of Quentin Tarantino's Obsession.